This is Mitosis by Ava and Gwen. So mitosis is a form of cell division that helps your body grow and repair any damage. The first step of 4 is prophase, where chromatin condenses into chromosomes. Chromosome is basically just an unwound chromosome. The nuclear membrane breaks down, releasing the chromosomes into the cytoplasm. The nucleus then disappears and the centrioles move to the opposite sides and/or poles of the cell. As they move apart, a network of spindle fibers form between them. Spindle fibers are composed of microtubules, which are hollow tubes of protein that control chromosome movement. So here we see the phases of prophase that happen in order: the condensing of the chromosome, the formation of the spindle fibers, and the disappearance of the nuclear membrane, and then the centrioles separating and moving to opposite poles of the cell. In step 2, we have metaphase. The spindle microtubules attach the chromosome's kinetochores and move them to the center of the cell. Centromeres are aligned on the equator. During anaphase, sister chromatids split from the centromere and separate from one another, and the spindle fibers shorten, pulling the chromatids closer to their adjacent side of the cell. One complete diploid set of chromosomes is gathered at each pole of the cell. In telophase, the cell begins by uncondensing the chromosomes and unwinding them back into chromatin. The spindle fibers break down and a nuclear membrane forms around.
Meiosis by Ava and Gwen. Step 1. Prophase. The homologous chromosomes condense and become visible. They pair up to form a tetrad, and they exchange genetic material by crossing over. Step 2. Pre-metaphase or metaphase. Microtubules attach the chromosome's kinetochore. The nuclear envelope breaks down, and tetrads line themselves up on the metaphase plate. The homologous chromosomes pair up and orient themselves randomly. Step 3. Anaphase 1. The centromere breaks down, and the homologous chromosomes separate. Step 4. Telophase and cytokinesis. The chromosomes move to opposite poles, and cells separate into 2 new haploid cells. Step 5. Prophase 2. The centromeres condense again. Centromeres that were duplicated during interphase 1 move away from each other to opposite poles, and new spindles form. Step 6. Pre-metaphase 2 or metaphase. The nuclear envelope breaks down. Each sister chromatids are condensed again and aligned at the equator of the cell. Step 7. Anaphase 2. Sister chromatids are pulled apart by the kinetochore microtubules and moved to opposite poles. Step 8. Telophase 2 and cytokinesis. Chromosomes arrive at opposite poles. They begin to decondense, and the 2 cells divide into 4 unique haploid cells.
The comparison between mitosis versus meiosis.
Mitosis produces 2 new diploid cells from a parent diploid cell, and meiosis produces 4 new haploid cells from a diploid cell.
Meiosis 1 is reduction division, but mitosis and meiosis 2 are not, because the number of chromosomes is the same as the parent cell.
In meiosis, homologous chromosomes pair up, cross over, and line up on the metaphase plate. In mitosis, there are no homologous chromosomes.